Hello, this is Module 5, video number 5, Extensions of Mendelian Genetics. In this video, I'm going to be talking about organelle genes, specifically mitochondrial genes, and examples of a couple of mutations that cause human disease. So those are called Leber's hereditary optic neuropathy, LHON, and myoclonic epilepsy and ragged red fiber disease called MERFF. First, let's take a look at the human mitochondrial genome shown on this slide. So this is about 16 or 17 kilobases circular genome. And this slide shows mutations that fit into two categories: those affecting the process of protein synthesis in mitochondria. And there are 153 known mutations. The other category is mutations that affect polypeptides that are encoded in mitochondrial genes, and there are 108 known mutations for those. So for the example I chose, LHON, Leber's neuropathy, is an example of a mutation in a peptide protein coding gene. And MERFF is an example of a mutation in a gene involved in protein synthesis. So let's look at LHON first. So LHON is due to a mutation in the gene, mitochondrial gene encoding the enzyme NADH dehydrogenase. This protein enzyme is necessary for ATP synthesis in mitochondria. People that have this disorder can be homoplasmic for a hypomorphic allele or heteroplasmic for a null allele. And just to remind you, homoplasmic for a hypomorphic allele means that they inherited from their mother cells that have only mutant mitochondria. However, they're able to survive and produce ATP because the enzyme is still functional, hypomorphic. It's a partial loss in function. The enzyme operates more slowly, but it's still functional. And they can produce ATP, just not as high levels as normal. For people that are heteroplasmic for a null allele, that means they inherited mitochondria from their mother, some of which are wild type and fully functional, some of which have a null allele and not functional. So they have a mixture of both wild type and mutant mitochondria. Notice that someone who inherited a null allele and was homoplasmic, meaning all mitochondria had this null allele, they would not be able to produce any ATP, so that's not a viable condition. The disease causes optic neuron neuropathy, and the optic neuron is what connects the eye to the brain, and it's carrying, optic nerve. It's carrying signals from the eye, the retina of the eye, that are processed in the brain, giving you visual input. What happens in Leber's hereditary optic neuropathy is that the optic nerve degenerates. Nervous tissue is extremely sensitive to deprivation of ATP, and the optic nerve is probably the most sensitive. On the right side, it shows how the disease progresses. Somebody with Leber's has progressive loss of vision. Now, this being a mitochondrial disorder, it's inherited by maternal inheritance. And this pedigree below is demonstrating that. So if there is a woman who has the disorder, she will pass it to all of her children because they are inheriting their mitochondria from her, not from their father. If a son gets Leber's, he will not pass it on because all their children will get wild-type mitochondria from his partner. If the daughter has Leber's, she will pass it on. So this is maternal inheritance. Okay. Now, if somebody inherits a homoplasmic for a partial loss of function, hypomorphic allele, the symptoms are pretty standard among individuals with that condition. But if somebody is heteroplasmic for a null allele, the symptoms can vary quite a bit. And the variation is due to the fact that different individuals might inherit from their mother a more or less severe condition. More severe would mean a higher percentage of mutant mitochondria. Less severe, a lower percentage of mutant mitochondria. All right, let's look at the other mitochondrial disease that I mentioned, and this is myoclonic epilepsy and ragged red fiber disease, and I'll call it MERFF. MERFF produces a range of symptoms. So the myoclonic epilepsy involves epileptic seizures with uncontrolled muscular contractions and kind of jerking movements. This disorder is due to a mutation in the gene for mitochondrial tRNA. And mutation is always null, wiping out mitochondrial protein synthesis. So this affects all genes encoding proteins in the mitochondria. It has a very drastic consequence, and that is it stops mitochondrial ATP production. So because this is a null allele which wipes out mitochondrial protein synthesis and therefore ATP production, it's not possible, it's not a viable situation to be homoplasmic for the null allele. So therefore, only people that survive with MERFF are heteroplasmic for this null allele. Now, the severity of MERFF varies due to the degree, the percent of individuals, percent of mitochondria that are mutant in various individuals. This slide is illustrating a cross-section through muscle tissue, skeletal muscle tissue. And one of the features of MERFF is that muscles produce a lot of mitochondria. Because mitochondria are not functioning, at least some of them, muscles tend to produce even more mitochondria as a compensatory reaction to less function of mitochondria in general. So if you stain muscle tissue in an individual with MERFF, stain it with a dye that specifically stains mitochondria, you'll see something like this. Compared to wild type, there's a heavy staining for mitochondria. And people with MERFF, their muscle fibers produce a huge number of mitochondria to compensate for lower function of each mitochondria. And the dye is red, and that's where the term ragged red fiber comes from, staining extensively with mitochondrial specific stain. MERFF shows maternal inheritance, just like the case for Leber's neuropathy. If a woman has the condition, she will pass it on to all of her children, and her son here will pass it on to none of his children. The mates are not shown in this pedigree, but they're implied. This also shows that the symptoms vary in intensity. So purple is extremely, are probably the most severe. Blue, less severe. Yellow, less severe, and blue, the least. And that correlates with, if you look at the percent of mutant mitochondria in cells from these individuals, it correlates with the percent of mutant mitochondria. Higher percentage, more severe disease. Okay, this also shows that from a single woman with the disorder, she can have children with variations in the extremity of the disease. So her daughters here all got cells with greater than 90% mutant mitochondria. Her son here got around 90%. So it varies, and that is due to the fact that during production of her ova, some ova got more mutant mitochondria and some got less. This last slide here shows that the different tissues that are affected by MERRF. And remember, MERRF is going to have mitochondria that are producing less ATP, or at least producing it less efficiently. So MERRF produces symptoms in the central nervous system, the heart, skeletal muscle of two different types. Type 1 is more ATP demanding than type 2, and in skin. And then this is showing along the left individuals that differ with respect to the percent of mutant mitochondrial DNA. Okay, so in this individual, 20% of the mitochondria have mutant DNA, 80% wild type. Here, 40% mutant, 60% wild type, right? So this is increasing percentage of mutant mitochondria. At low levels of mutant mitochondria, only 20% of individuals with that type of heteroplasmy typically only show symptoms in the central nervous system, the brain. So that would be the myoclonic epilepsy. Individuals with 40% brain and then start to see heart defects as well. 60% brain, heart, and the more energy demanding type of skeletal muscle. 80% you start to see symptoms affecting all types of tissues. So individuals can vary in the percent of mutant mitochondria, and that produces different types of symptoms as a result. Now, recently a technique was developed to allow mothers that have a known mitochondrial disorder to have children that don't have this disorder. Typically, mothers will pass it on in their mutant mitochondria to their offspring. But this technique involves using a donor egg from a healthy female that doesn't have the mitochondrial disorder and harvesting an egg from her, removing the nucleus from her egg, and taking the nucleus from the mother's egg and implanting into the donor egg. Then this donor egg is fertilized with the father's sperm in vitro, and that embryo is implanted into the mother. Okay, and this slide is just showing the actual process of removing the mother's nucleus and implanting into the donor's egg, from which the donor's nucleus has been removed. So most of the genetic material in this egg is coming from the mother, but we're avoiding using her mutant mitochondria and instead relying on the normal wild-type mitochondria from the donor egg. So this technique is called oocyte nuclear transplantation, and this was first used successfully in humans in 2016. So I'm going to end the video here, and the next video we'll talk about extensions of Mendelian genetics for traits determined by 2 genes. See ya.